Okay, so good afternoon or good evening. So let's get on with our immuno. So what I'll do first is I'll just quickly, run through, just the picture, the big picture of all the important events that we covered so far. And then as I do each step, I'll just tell you what are the kind of questions that you could probably, expect from each of these events. And then we will go over some of the questions that I have for you. Okay? So obviously we will start with the invasion of a microbe, because that's where everything begins, isn't it? So let's think of a bacteria, or it could be a virus. I'll call this a virus, and this is a bacteria. Okay? So this is invaded, it's already entered the system. So now what we need to think is, what would be the first, what event or which cell is going to meet this microbe? Good, a macrophage. So let's draw a macrophage here. This is a macrophage. Okay. What else would be lurking around? A dendritic cell. Yes, so we have to, the dendritic cell. Anything else? Any other? Okay, no, I'm not talking about cells. Anything else that would be not cells? Anything else that you would think of which would play a role in the innate defense that you should not forget? Complement. Don't forget the complement proteins. Okay? The complement proteins also will be around here somewhere. So I'll just put them as C, C, C. Okay? So now, the macrophage will obviously recognize that something is wrong. How is it going to recognize it? What is there on the surface of the macrophage? The toll-like receptors. Yes, don't forget the toll-like receptor. Okay? So the TLR, which is also called, what is it also called? PRR, isn't it? Pathogen recognizing receptors. The dendritic cell will also have TLRs on their surface. Okay? So the TLR is going to be the first receptor that is going to recognize a pathogenic pattern. Okay, so if it's a bacteria, it will recognize a pattern on the bacteria. If it's a virus, what part of the toll-like receptor would recognize what of the virus? Do you remember? Where are the toll-like receptors for the virus located? Inside or outside? It's inside the cell. So the virus has to infect the cell. The virus has to infect the cell. And when the virus infects the macrophage, just wanted to, just making it, yeah. So when the virus infects the macrophage, there are toll-like receptors inside. So why I'm emphasizing this is because you must not forget the toll-like receptors. Okay, so the TLRs are present inside and outside. Any microbe that's intracellular would be recognized by the toll-like receptors inside. These are the first receptors. So this is an important TLR. There have been even board questions on the toll-like receptors. Okay, so that's why don't forget the toll-like receptors. Then the complement, which is floating around here, what is the complement going to do? What would the complement do first? Which complement pathway is going to be activated? The alternate pathway. So some of the which complement component then would recognize the, and what is it going to recognize? The C3. So you have the C3, which is spontaneously generated, will go and stick to the surface. You see? I won't show it as C3b, sorry, C3, C3. So remember the C3, of course spontaneously the C3 is cleaved and they will go and coat the microbe. So this is the alternative pathway, because that component, the C3 component, can recognize that's a trigger, isn't it? So that's the first pathway to be operated. So that is going to coat the microbe, and that would help, that will initiate the alternative pathway. So what you need to remember is that, so here, boom, boom, you know, you're going to have the cascade going off, the complement cascade going off. Okay, so what is going to be generated in this cascade? C5a, C3a, C5a, C4. This is the alternate pathway, isn't it? So no 4, isn't it? This is the alternate pathway. Okay, 3, 5, the MAC complex, but 3a and 5a is generated. Then, so this is happening here. We are concentrating on a bacteria. Let's leave the virus aside. What's a macrophage going to secrete now? Yes? Interleukin 1, interleukin 6, TNF alpha. Good. Okay. So now these cytokines, so now you have a dendritic cell lurking around. The dendritic cell doesn't want to miss anything of the show. I mean, what's going on there. So the dendritic cell will also now sample a bit of the pathogen, antigen, because, you know, this is the postman. It has to travel and take the antigen and inform the adaptive immune system. So you see, now what's going on is the innate immune system. Okay, so now these cytokines, what is it going to do to the dendritic cell? The dendritic cell is in the milieu of these cytokines. The dendritic cell also has its toll-like receptor also recognized. The toll-like receptor also recognized. Okay, so what is the dendritic cell going to do now? In response to these cytokines, what will it do? It will upregulate, yes. So now the dendritic cell matures, and first it will upregulate its MHC2 on the surface, and then it will also upregulate B7. B7, which is the co-stimulatory molecule on its surface. Got it? Because it sampled a pathogen, it has to let the T lymphocytes and B lymphocytes know that I have a pathogen here, so be warned and be ready. Okay, so it's upregulating the co-stimulatory molecules. Now remember, there are some dendritic cells and macrophages which are also phagocytosing. Macrophages' job is to scavenge. So there could be lot of proteins and other antigens, protein antigens, which are harmless. Every day we are exposed to so many different protein antigens, you see. They are also phagocytosed by the microbes. The dendritic cell will also sample those. They will take it to the, you know, lymph node and spleen, but that will not cause the upregulation of the co-stimulatory molecules because they don't come from a pathogen. The dendritic cells, it's a harmless protein. They have swallowed it, they've eaten it, but it is not going to upregulate the, you know, B7 and MHC2 much co-stimulatory molecules because its toll-like receptors did not recognize a pathogen-associated molecular pattern. Is that, now does it make sense? Okay, so now even the bacterial surface, you see there are some bacterial surface proteins also which can, you know, stimulate the upregulation of the co-stimulatory molecules. So now you see how the inflammation is going to start. You have the inflammatory cytokines, you have the anaphylatoxins which are released from the complement. Now this is going to bring about, so you have these, you know, our neutrophils, they are floating around here, neutrophils. So what are these cytokines? What changes in the capillary? This is a capillary. So what's going to happen now? These cytokines will diffuse, these will diffuse. What's going to happen now? What will they induce? What will they induce on the endothelium? This is the capillary endothelium. What are they going to induce on the endothelium? The selectins, yes. So you'll see sprouting here. Yes, P-selectin and E-selectins. Okay, so they are going to sprout here, P and E-selectin. So now the neutrophils, when they see a lot of P and E-selectins, the Cialis-Lewis residues on the surface of the neutrophils are going to attach to the selectins. So they are rolling, they're not rolling, they were floating at high speed, suddenly they stop and they all marginate. They go and stick to the walls of the endothelium, so they are slowed down and they keep rolling on the walls of the endothelium until, of course, what is the big one? The LFA, yes. So the LFA integrin, LFA1, on the surface of the neutrophil, then you have the LFA1, sorry. Okay, so these, they were in this state. They will open up like a switchblade, so that is in the high integrity stage, and then they can adhere to its, ligand on the endothelium, ICAM. Okay, so first the P and E selectin will, help them to marginate, and then the LFA1, which is upregulated on the surface of the neutrophil, will help it to squeeze through. In fact, it will help it to squeeze through the walls of the endothelium and go here. Okay, and then of course it has so many of its granules which will be secreted, and you will see inflammation. Okay, so this in a nutshell is what is mainly happening here. So usually they test you on what is the toll-like receptor, what are the cells that play an important role in innate defense. As you can see, the macrophages and the dendritic cells, that's why they are always patrolling. And the neutrophils arrive, they are the first to arrive at the site of the infection. Why the confusion? Because of the arrival. The macrophages and dendritic cells are already there. How do the macrophages originate from? Monocytes. Monocytes, when they mature, when they become, in the tissues, the monocytes mature into macrophages. So tissue monocytes are macrophages. Okay, so the complement. Now suppose the microbe here, the microbe had mannose on its surface. Okay, if there were mannose residues on the surface of the microbe, then the lectin pathway would have been activated. There are some fungus and some microbes which have those molecules on their surface. So the lectin pathway, what's the first component of the lectin pathway? Yeah, it starts with C4. So the mannose binding lectin will bind to the mannose residue and trigger the complement pathway. Okay, so alternative and the mannose binding lectin pathway could be triggered here. Now the dendritic cell will travel all the way with the antigen. It has to go to the lymph node and the spleen. Okay, so if this antigen was an extracellular antigen, most likely it would go to the spleen. The extracellular antigens will go to the spleen, and most of the intracellular will go to the lymph node, because the lymphatics will drain it. So now here, so now you see how the innate cells of the innate immune system actually triggers, they are the first to initiate the adaptive immune system. If you don't have macrophages, if you don't have dendritic cells, the adaptive immune system is not going to be stimulated. So they act as a bridge. They act as a bridge between innate and adaptive immune system. Okay. So now, before that, let us just briefly look into the antigen presentation. Now this antigen presenting cell will have what, which are the molecules on its surface? 1 and 2. Because it's a nucleated cell, it will have 1 and 2, but if it has phagocytosed the antigen, it will display it in, if it has phagocytosed, if the dendritic cell has phagocytosed it. Yes, if it is phagocytosed, remember it will be enclosed in an endosome. Okay, so then it will be displayed in 2. The antigen will be displayed in 2. Okay, so the dendritic cell now will take it to the spleen as well as maybe to the lymph node. And who is waiting there? Who's waiting in the lymph node and spleen? You have the naive T cells and the naive B cells. B cells with the T cell with its receptor. Okay, so the dendritic cell will now take it to the lymph node and spleen. Now briefly, which areas of the lymph node are you likely to see the T cells? T cells in the paracortex, B cells in the cortex. Okay, and in the spleen, periarterial, yes. Surrounding this would be the T cells, T cells in the sheath, and the germinal centers are also right here. Okay, B cells in the germinal centers. Okay, so now the dendritic cell has traveled all the way and it will enter the lymph node through high endothelial venules, and these cells will just pour into the lymph node, and there, depending on the cytokines, they will migrate to the T and B lymphocyte, the cortex and the T and B cell areas. Okay, now here again the same adenosine molecules like the L-selectin, the LFA1, they play a role in the migration of the T lymphocytes. So the T and B lymphocytes would have come here from the bone marrow, T lymphocytes would have come here from the thymus. Okay, so the markers and everything we will just see a little later. So now the dendritic cell will reach there and it will present the antigen, and the activation of the T and B lymphocytes will begin. Okay, so up till here, these are the more, usually they will ask what are the what are the important complement components. So we will just compare it with the, there are a few questions, so that time we will clarify the activation of the complement. So just briefly, you know that the developmental stages of B lymphocytes, I won't, with the questions we will just look at that, so I'm not going to go into that. So there you have the T and B lymphocytes, and as you saw yesterday, depending on the antigen, if it is, let's look at it this way, you have the extracellular antigens, okay, and you have the intracellular here. Now, in the extracellular, antigens are dealt with by B lymphocytes. Okay, so you have a B lymphocyte with, and here you have another B lymphocyte also. Okay, so if this is a non-protein antigen, if it's a non-protein antigen, what's going to happen? What's going to happen? IgM, plasma cells, it will differentiate into a plasma cell which will secrete only IgM. Stop. Okay, if it is a protein antigen, this is a protein, the B lymphocyte will process it because this is an antigen presenting cell. It has MHC2 on its surface. It has, it will present it to the CD4 cell, TH0, and then this will differentiate into, so with this help, this is first going to differentiate into a plasma cell, which will secrete what? IgM. Then the T lymphocyte will differentiate into what? Th2. Okay, what is the cytokine? IL-4. Then this starts secreting more IL-4 if it is IgE that you want. It will secrete TGF-beta if it is IgA that you want. And some interferon gamma, depends on the antigen. Yes, interferon gamma will isotype switch it to IgG. Okay? Okay? So this is how the Th2 will help. And then of course other cytokines like interleukin 5 will call the eosinophils. That all depends on the antigen here, what the antigen is. Okay? Whether it's a worm or whether it's an allergen. Okay? Now if it was an intracellular, once again this could be inside a macrophage. There could be a dendritic cell here which is also presenting this antigen. The dendritic cells also can present remnants of the intracellular microbe to a CD4 cell. So how will the CD4 know that it should differentiate into Th1? How will it know? What does the dendritic cell? Yes. Yes. It is IL-4, yes. I don't, yes. Okay, yeah. So, the dendritic cell is now going to display this antigen which it managed to phagocytose again with two CD4 cell. And what is it going to secrete to tell the CD4 to differentiate into Th1? Interleukin 12, good. So, interleukin 12 secreted, CD4 differentiates into Th1. Th1 will secrete interferon gamma. Now there are some natural killer cells also. They also will respond to interleukin 12. Okay, just natural killer cells also respond to interleukin 12 and secrete interferon gamma. Okay. And the interferon gamma acts on the natural killer cell and stimulates it. So that's separate. So now the interferon gamma will cause more of CD4 cells to differentiate into Th1. So this goes on like a cycle. Now this interferon gamma will go all the way and help the macrophage to kill. Yes, it now activates it and then the macrophage can kill the pathogen. So the antigen presenting cell here again is helping out the macrophage to, antigen presenting cell is again presenting the antigen to CD4 and the CD4 is helping out the macrophage by secreting interferon gamma. At the same time the CD4 will also secrete interleukin 2. Wherever there are proliferation of CD4's T lymphocytes, interleukin 2 is a very important cytokine. Proliferation, activation and proliferation is by interleukin 2. Okay, so what another point that you need to remember is this cytokine can inhibit Th2 and interleukin 10 that is secreted by 2H2 IL-10 will also inhibit interferon gamma. Okay. So interferon gamma will inhibit the production of IL-10. IL-10 will inhibit the production of interferon gamma. That way one of the pathway is kept suppressed. It's not that it's completely absent, but that should not be dominant. It is the suppressed pathway. Okay. So here what is always tested are the cytokines. Every cytokine that is produced here, its function is important. They keep testing you on the cytokines. So far we have seen starting from interleukin 1, What is its function? It's a pro-inflammatory cytokine and it normally induces fever. Then interleukin 2, activation of T lymphocytes. Interleukin 3, myeloid. Okay, so it's produced and it differentiates into myeloid. Interleukin 4, it's a Th2 cytokine, isotype switching to IgE. Interleukin 5 activates eosinophil and it's supposed to enhance the production of IgA according to, just remember that also. But mainly it's an eosinophil activating cytokine. Differentiation of, then interleukin 6, it's also a pro-inflammatory cytokine. Yes, it helps in isotype switching. And interleukin 6 also mainly activates the liver to produce acute phase reactants during an inflammatory reaction. Interleukin 7, lymphoid, yes. Interleukin 8, neutrophil chemotactic agent. Then we don't have 9, 10, produced by Th2, inhibits the Th1 pathway. So it is also known as a anti-inflammatory cytokine because it inhibits Th1. Then interleukin 12, it's also a Th1 cytokine produced by antigen presenting cells and also produced by, in this picture, macrophages. Macrophages also will produce interleukin 12, which can, the circle continues. Okay, so interleukin 12 is produced by antigen presenting cells. It is also produced by macrophages, interleukin 12. Okay, and natural killer cells also will produce, macrophages produce interleukin 12, Th1 produces interferon gamma. Okay, then Th17, it's a pro-inflammatory cytokine, mainly stimulates production of neutrophils. Then interleukin 17, after that 18, 19. That's about it. Those are the major cytokines. Yes. Where? Oh no, that is the interferon gamma that is produced by Th1. That interferon gamma also helps in isotype switching to IgG because the IgG antibodies are required for opsonization by the macrophage. So you see, that's connected. So to help the macrophage, okay, yeah. So yeah. IL-6 also has a role in isotype switching, but mainly it stimulates the liver, acute phase reactants, mainly, okay? And interleukin 6 also acts with interleukin 1. Sometimes it's also referred to as a fever inducing, but it's mainly IL-1. Which one? IgG. Interleukin 6 can to some extent cause isotype switching to IgG. Okay, that's one of, so one point that you should remember is you would now see that the same cytokine can have multiple function. So cytokines, sometimes they are autocrine, they will, the same cell which produces it, it acts on the same cell. It can have multiple functions too. The same cytokine can have more than one function. So these cytokines and the function are frequently tested, and you must remember them in the context, otherwise they don't make any sense. Okay, they're just numbers. So now let's look at some of the questions, and then wherever you have a doubt, we will I will clarify it. Now you can use the clicker if you want, but just to know, I'll just give you a few seconds to Okay, just let me just see. Okay, so what does this look like? So which is the first molecule, what's the first receptor? It's a toll-like receptor, which is also referred to as PRR. Okay, so just for you to remember this, it's a toll-like TLR or PRR, recognizing PAMPs on the microbial, the pathogen. Okay, so that's so you got that. So here's another one. Oh, I need to close this end. Okay, let's see. So these two toll-like receptors, what do they bind to normally? 3 and 7, the odd numbers, they bind to viruses. Okay, the just at random, just remember the even numbered ones. You see the 2, 4, 1, 2, 4, they bind to bacteria. Just at random, we just wanted to. I'm not going to ask you the numbers, but just for you to know that these bind viruses. Okay, so obviously this child will have increased resistance to viruses. Eventually in your comp and board you have to know the numbers, but I'm not going to ask you the numbers in your mini because I don't want to stress you too much. Okay, okay. This is an easy one, but it's very, very commonly asked. Done? Oh, good. I'm happy. Yes. Yes. Okay, so 1, 6 and TNF alpha, all of you know these are the, and please remember that these cytokines have so many effects as you saw, okay? So you see it will upregulate the, selectins and on the surface of the endothelium. It upregulates the co-stimulatory molecules on the surface of APC. Don't forget all that, okay? So these cytokines are very important, 1, 6 TNF alpha, usually the septic shock, and these are mostly stimulated by bacterial cell wall components. Okay, so that's why bacterial infections often lead to shock. Okay, good. Okay. This requires a little bit of logic, just logic. Okay, so which one of the complement components do you think would have a major impact on all the three pathways? Good. 3. Because you see, if you look at the classical pathway, it starts with 1, 4, 2 and 3. Okay? Alternate starts with 3. Mannose binding lectin 4, 3, no, 4, 2 and 3. Okay? So now if 3 is absent, everything comes to a standstill. You see the classical will start at 3, stop. Alternate will not start at all. And mannose binding lectin, when it comes to the level 3, yes. Pardon me? Okay, so if it starts with if it's case of a mannose binding lectin, when it reaches 3, stops, isn't it? So when you're faced with a question like this, remember that 3 is the most crucial component of the complement. If 3 is defective, all the 3 pathways will stop. There will be no complement activation. It may be activated, but it comes to a standstill. It cannot complete. You see, the cascade is not completed. The MAC is not formed. Maybe some anaphylatoxins may be generated, but even the most important component, C3b, which is required for opsonization, even that is not going to be generated. Okay, so remember, this is, so if it is a deficiency of C3, then even the other components would be missing. The ones after this also will not be activated. Okay, so the next one, I'm sorry, it just happened. I was trying something. Okay. So what is the cell that they're referring to here? The NK cells. Good. So which of the profile matches the NK cells? It has to be CD3 minus, CD16 and CD56, isn't it? Because natural killer cells will not have a CD3. It has 16 and 56. So remember, natural killer cells would always target, normally it would target a cell which does not have MHC or in which the MHC is down-regulated. Yes? Yeah. It doesn't need a signal transducing. It does not have a TCR. Okay? It doesn't have this. It doesn't have CD3. NK cells don't have CD3. Negative. CD3 negative. Yeah. Okay. Okay. This is also a commonly asked one, but I know you know what the answer is. Done? Okay. So what is this? What's a defect? It's a chronic granulomatous disease because the child is getting recurrent infections with catalase producing bacteria. So this is because of a deficiency in NADPH oxidase. So if there is a deficiency in NADPH oxidase, the other thing that you should remember is, what about the nitro blue tetrazolium test? Will it be yellow or blue? It will be yellow, okay? So the nitro blue tetrazolium test, if the NADPH oxidase is defective, it doesn't get the blue color, it stays yellow, okay? So this is a deficiency of NADPH oxidase. If there is a deficiency of myeloperoxidase, what would happen? Yes, all bacterial infections with catalase positive and catalase negative, everything will, all of them will has an equal chance, and the child would of course present with abscesses, but the NADPH oxidase would be normal, okay? Myeloperoxidase is deficient and the child will have skin infections, recurrent skin infections, okay? So the other options are not as appropriate as the NADPH oxidase. So go for that. You know this one. So this is, 7. So I won't spend too much time on this, but just showing this because this is so commonly asked, okay? So lymphoid cell is 7 and another 100%? No, 94. Okay. So lymphoid cell is 7 and myeloid is 3. So I'm skipping the next one. The next is myeloid. So all of you know that it's 3. Same question. Okay, let's go to the next one. This also is easy. Oh, this is, this is, it should have been, no, there's something wrong with this question. Let's skip this, typing, yes, no. Next one, okay. Okay. Oh. So there is a deficiency, decreased number of lymphocytes in the paracortex. So which subset of cells are going to be the T cells? Now the question is asking you about which cells would be normal in the periphery. So what cells are you going to see in the peripheral blood? The B. The B is unaffected. It's a T that is affected. Okay? So don't get, they ask the question exactly like this. So don't get caught. Here they're asking you about the normal cells, but they're just twisting it and asking you, okay, so peripheral blood will have normal to elevated. They are not going to have much T lymphocytes, but the B lymphocytes are unaffected because there's some problem in the paracortex. So this is just to test, to see whether you know which cells are populated. So you see CD3 is a T cell, C is a T cell, 4 is a T cell, this is a T cell, this is a NK cell, and this is also a T cell, okay? So here the question is lymphocytes. So natural killer cells Natural killer cells don't fit into that. Mainly lymphocytes is for T and B. Okay? Then it's an easy one. So where are the tissue antigens normally drained into? Tissue antigen would go to the tissue antigens would go to the lymph node. Okay, the lymph node tissue generally drains to the lymph node, and the blood antigens drain into the spleen. Okay, that's generally the rule. So they like to ask this just to know whether the lymphatics normally take the tissue. The nearest draining lymph node, it usually goes there. Okay, so this would be the lymph node. Okay. Okay. Good. So, what do you think is the right answer? E. Why? Yes, the main clue here is, don't miss this, it's a TDT positive, okay? Because you can see pre-cells with the cytoplasmic and the mu, which is either in the cytoplasm or on the membrane at the end of the pre-cell, the beginning of the immature cell. But if TDT is positive, that means a rearrangement is still going on. So you can't have something, you know, on the surface or in the cytoplasm, okay? So that's why keep a, you know, note, look out for the TDT. If that is positive, that indicates that the arrangement is going on, okay? Fine. So this is the right one. Okay. Yes, so what's the cell that they're referring to? B cell. So remember, CD19 is always the most topmost marker for B lymphocytes. So when you see that, ignore everything else and go for the CD19. Okay, so here they're referring to a B lymphocyte because they're mainly calling the cortical area. Okay, So know this well, because you're very fond of asking this, cortex, paracortex, and the type of markers on the cells, okay? Okay. CD19. Oh, this is another one. I think we'll skip this. You know this. We just, yeah. Okay. The A is missing, but okay. Hmm. Hmm. Okay, so what is that blue region? It's the, yeah, it's the cortex, and that's the follicle, isn't it? Primary follicle, where the B cell interaction takes place. Now, so here they're referring to now which of these processes is likely to take place there? Let's look at each one of them. VDJ recombination, where does it take place? Does it take place in the lymph node or in the bone marrow? Bone marrow. VJ recombination, bone marrow. Negative selection, thymus or bone marrow, okay? And tolerance induction, same, in the bone marrow or thymus. So all these are processes that take place before encountering an antigen. Isotype switching is after encountering the antigen. An antigen and antibody encounter, or antigen and lymphocytes encounter always take place in the peripheral lymphoid organs, okay? So this is an important concept which you should remember, okay? Isotype switching, yes. Yeah, after they meet with the antigens, yes, they, mostly the plasma cells are usually mostly seen in the medulla. Isotype switching can happen, that's already differentiated plasma cells. They have already differentiated plasma cells. Yeah. Yeah, plasma cells differentiate there in the medulla. Anyway, what isotype switching can take place even in anywhere in the lymph node. But the plasma cells, yes, they predominate. Okay, whatever it is, even if you have a doubt about that, definitely you know that only isotype switching takes place in the lymph node. Nothing else, none of the others can take place in the lymph node. That's what they want you to know. Okay? Yes. All the other options are definitely wrong. Okay. Okay? Hmm? Okay, so what's this? So you have, in addition to small changes in the B cell DNA encoding the variable region of the light and heavy chain. So what is this change when there is a, after encountering an antigen, what is that called? Somatic hypermutation. And there is also a change in the constant portion of the heavy chain. What would that be? Isotype switching, isn't it? Both these events can take place after, uh, yes, the second encounter with the antigen. So when IgG antibodies are produced, there is an isotype switch, and the antibodies binding region will have a better affinity. Okay, so these are two changes that take place after the encounter with the antigen. Yes. Is there any other option with isotype switching and somatic hypermutation? No. Yeah, okay. What you're saying is that the chronological order, the sequence. No, but then there is no other option which has the both, isn't it? So, pardon me? Receptor editing is what happens in the bone marrow. You see those B lymphocytes which have a strong affinity to self-antigens, they are usually eliminated by negative selection. But some of the B lymphocytes get a second chance and they can alter their receptor. That is receptor editing. Okay? Yeah. So, is that okay? Yep. Okay. Oh, this one is, yes, this is. Okay, this is an easy one. Where are you likely to find this? In the cortex. What did you think of? God. Thymus. It's, where would you see this double positive? Where does the selection take place? They're double positive cells, CD4 plus, CD8 plus. They're double positive cells, CD4 plus, CD8 plus T cells. It's not CD4T, CD8T. Okay? So where will you see them? Where will you see them? You'll see them in the thymic cortex of the thymus. You see them in the thymus first and foremost. And then you see them in the cortex of the thymus. So most importantly, please know that you see them in the thymus. You can't see them anywhere else. Isn't that where the markers are acquired? Pardon me? Where do you see the where's paracortex? That's in the lymph node, paracortex. This is thymus. Okay, these are immature T lymphocytes. They have not even matured yet. So they are the double positive, the first stage. They get both the markers and they are still in the thymus. Okay? Please know that you cannot see double positive cells in the peripheral lymphoid organs. If you see it, there's something wrong. This is also, you know this. That's the easy one. Oh, I didn't. So if there's no CD3, what's the importance of CD3? Signal transduction. That's absolutely essential for the survival of T lymphocytes. That's the first of the markers that a T lymphocyte needs. Okay, so if there is no CD3, there is no survival. They die. So remember that if there is no CD3, there is no ability to signal transduce or to proliferate in response to an antigen. Okay, so that's the answer. Okay, this is the same. Leaves the bone marrow. Oh, I should have done this. I'm sorry. What's the answer? Huh? Double negative and nothing. They arrive naked to the thymus. They have nothing on their surface. Okay, so they are double negative, TCR negative, because it's in the thymus that all the rearrangement and everything takes place. Okay, so remember, when they leave the bone marrow and they reach the thymus, they have nothing on their surface, not even a T cell receptor. Okay, so this is the MHC1 means peptide and MHC1. This is peptide MHC1. Okay, this means peptide MHC1. Okay, I think I can understand why the confusion is. Okay. Okay, so what do you think is the what happens to it? Do you think the cell will survive? No. So even though it has undergone positive selection, still if it binds strongly to a peptide MHC1 on the epithelial reticular cells, it's again a self MHC. So if it binds strongly, it will get eliminated. Okay, so no T lymphocyte should recognize a self. See, it's not an antigen presenting, it's not displaying a foreign antigen. So it survived a positive selection, but again somehow it's binding strongly to an MHC1 with a self peptide. So these cells will get eliminated. Anything that binds strongly to a self antigen would normally be eliminated. So it's a negative selection. Okay? Pardon me? Because it is on the epithelial reticular cells. They are not, yes. Yes, that's how sometimes they denote. P means a peptide is there in the MHC1 groove. Okay? It survived. There is a pardon me? It has survived, but again it goes through another round. So even after it survives, if it binds efficiently to a self MHC, a peptide self antigen, it will be eliminated. There are multiple rounds. So here the point is, it does not indicate in any way that it's a foreign MHC. It just says from the epithelial reticular cells. Okay, so it's a second round of negative selection. In the thymus, negative selection takes place. There is central tolerance in the thymus, and even if some of the cells escape, there is another peripheral tolerance. So once in the thymus, sometimes twice in the thymus, then when it is outside again, it undergoes. So if it escapes the central and peripheral tolerance, then autoimmune diseases can result. Okay, so there are multiple rounds of selection. Yeah. Pardon me. It's dead. Negative selection means it's gone. It's dead. It survives if there is positive selection. So one round it survived. But then again it is binding strongly to the self antigen. So the second round it gets eliminated. You see? Because there are multiple levels of checking. Because sometimes they do escape one level. So the second level it gets. So here the thing is, it's a self peptide. Yeah. Receptor editing is in B lymphocytes. T lymphocytes don't undergo receptor editing. First time it did not. It somehow got positively selected. See, what I'm trying to say is there are multiple levels. It escapes the first time. See, not all cells sometimes will get eliminated. Our immune system has several levels because it's very crucial. So if it escapes the first level, at the second level, again if it binds, and again if it is binding too strongly, it gets eliminated. So there is central tolerance, then there is peripheral tolerance. There are multiple layers of checking. So self-peptide that binding should not happen. It should get eliminated. Okay? Yeah. Yeah. That is after antigen. This is this is the selection process. Okay? That what TB conjugate is when a foreign antigen is presented. Okay? That happens in the peripheral lymphoid organs, lymph node and spleen. This is in the primary lymphoid organs. This is the initial selection. You are eliminating self clones. They all have to go. Okay? Yeah. You had a question. They won't get selected. Anything that binds strongly should go. Yeah. They are positively selected. That's what it survived first. It did not bind strongly the first time, so it survived, but it was not it escaped. So it went through the second round and then it got eliminated. The point here is binding tightly. If it binds, if it has too much affinity to self proteins, it gets eliminated. It should go. None of the T lymphocytes which has got strong affinity should stay. Okay, they should go because they're dangerous. So see, the point that one of the points that you should try and understand is the MHC is always displaying our own peptide antigens, and there should be no T or B cells which recognize our own antigen strongly. They should go, because if they stay, it can lead to autoimmune disease. That's the purpose of negative selection. So any T lymphocyte which has weak affinity, because the T lymphocytes which have weak affinity are selected, that is why you have all those LFA, those integrin combinations between the, on the antigen presenting cell and the T lymphocyte. That is what keeps them together. Because they have weak affinity to the self MHC, only those remain. So what would be the response? It's a polysaccharide antigen, so the B cell will bind to which option? The C, yes. So this is a polysaccharide antigen, so the B cell will bind to the antigen and get activated independent of T cell help, because it is a polysaccharide antigen. Okay? Is there any confusion in this? Just give me a second. Wait, no, this is the old one. What does this look like? If there is a defect in the antigen presenting cell, will there be antibody production efficient? No. Will there be helper T cell activation? No. Will there be cytotoxic T cell activation? No. Will there be complement activation? Yes. Will there be macrophage activation? No, because you see the Th1 interferon gamma is needed for macrophage activation. Okay. This is also a commonly asked concept. Okay, let's see. Not bad. Okay, so what is this question asking you? This is about, yes, good, T cell proliferation. So you have a CD4 cell, which is actually a memory cell, which means that, what does it mean? It means that it has already been exposed to an antigen and it has been activated. But when this cell, he noticed that the incubation of CD4 cells plus just the antigen alone is not causing any proliferation, because what is the third signal? You remember there are three signals. What's the third signal? First signal is TCR recognizing the antigen. Second signal is the co-stimulatory molecules. What's the third signal? Interleukin 2. The third signal is interleukin 2. There is no interleukin 2, there is no proliferation of activated T lymphocytes. Now, who produces interleukin 2? Who produces interleukin 2? Activated T cells. And the interleukin 2 acts on the same T cells. So interleukin 2 is produced by activated T cells, and it acts also on the T cells. So when these are those experimental scenarios which they keep testing throwing at you because they just want to know whether you can figure out that here they're talking about proliferation. So please remember this, that unless there is interleukin 2, even though a cell is activated, unless it gets interleukin 2, it will not proliferate. It will just stay as one. You see, we want to get many T lymphocytes. Okay, so here the supernatant had interleukin 2 because it came from the activated T lymphocytes produced interleukin 2. The first one, the cell did not have, just the antigen, and the T lymphocyte did not have interleukin 2. So for proliferation, interleukin 2. So this is one of the very commonly tested interleukin, very commonly tested cytokine. And just keep this in mind. Two things you need to keep in mind, that T cells respond, only activated T cells respond to interleukin 2. Activation means it has already encountered an antigen. Okay? So this question is like a puzzle. It's actually simple if you just look at it. Okay? Okay, so a researcher is studying a specific immune response to antigen by incubating it with several cell types. He noticed that the antigen will cause activation of B cells alone, but not T cells alone. So one possibility could be that it is a polysaccharide, isn't it? Okay, but let's look at the next part of the question. If it was a polysaccharide, and it was with the antigen presenting cell, how did it stimulate the T lymphocyte? So what do you think it is? When it was incubated with the antigen presenting cell, then the T cell responded, isn't it? T cell, will it respond to a polysaccharide? No. Why did it not respond the first time? Does T cells respond directly to peptide and protein antigens? No. They have to be processed by the antigen presenting cell, broken down into peptides and then presented. So obviously, the first time the T lymphocyte was exposed to the protein. When it was mixed with the antigen presenting cell, the antigen presenting cell broke it down to peptides and presented it to the T lymphocyte. Okay? So it could have been a polysaccharide. If it was a polysaccharide, then the T lymphocyte will not be stimulated. You must remember that B lymphocytes can respond to unprocessed protein. B lymphocytes can directly respond to protein. It can respond to peptides. It can respond to polysaccharide. But T lymphocytes will only respond to either 20 amino acid peptides or 10 amino acid peptides, either by MHC1 or MHC2. Is that clear now? That's what this question is just testing and asking you. Okay? Okay? Huh? The answer is whole protein in native conformation. The first one. Option A. Yes? Yeah. Here the question is, first it was exposed to B cell and the same antigen was exposed to T cell. B cells alone in the sense without any antigen presenting cell. That's the antigen and the B cell. Antigen and the T cell. Okay? So it didn't respond. Then when it was mixed with the antigen presenting cell, the T lymphocyte responded. So from that, we should figure out that the antigen cell processed and presented it to, yes. A whole protein that has been denatured. Denatured proteins, no. Not a denatured one. Okay? So this is, okay, so this question is just to clear, you know, the concept of antigen presenting. Just for you to know that the B lymphocyte can respond to whole proteins or it can respond to polysaccharides. It can also respond to processed proteins. But the T lymphocyte will only respond to peptides that are presented by an antigen presenting cell. Okay? Yes. There are 2 dendritic cells. Okay, so this is an easy one. What's the answer? Huh? What's the answer? The T cells. There is a problem with the MHC class 2. So which are the cells which have MHC class 2 on their surface? Yes, B cells, dendritic cells, macrophages. T cells do not have MHC2 on their surface. They have MHC1. And therefore, T cells are not going to be affected. Okay? Remember, T cells have MHC1 on their surface. They don't use it, but they have it because they're nucleated. Okay? Okay, there's something wrong. You've seen this question before. This is not working. Yes. So, what's the answer? The answer is B, because antigen presenting cell will phagocytose and enclose them inside the endosome. The acid pH inside the endosome is required for breaking down the protein or the antigen. Okay? So if there is no acidification of the endosome, there is no processing of the antigen, and therefore it cannot present it to CD4 cells. Okay? So this is a very commonly tested concept. Please remember that antigen presenting cell phagocytose and process the antigen inside the endosome. Endosomes need acidic pH to function. If there is no acidic pH, there is no breaking down of the antigen. Okay? Okay. So the correct answer is B. Just give me a second. Let me just stop this and yeah. So the receptors for cytokines are synthesized only when it is exposed to the cytokine. Okay? So which of the cytokines would stimulate interleukin 12 production? Which cytokine would stimulate the production of interleukin 12? Which cells secrete interleukin 12? Which are the cells which secrete interleukin 12? Th1 cells and the antigen presenting cell. The antigen presenting cell will secrete, not the Th1. Sorry, antigen presenting cell will secrete interleukin 12 and that will cause the, what's the effect? CD4 to differentiate into Th1. Th1 will secrete interferon gamma. Interferon gamma will activate macrophages. Macrophages will secrete interleukin 12, which will again stimulate the Th1 cells. So, interferon gamma secretion would cause these cells to secrete interleukin 12. 12, interferon gamma 12. Please know it. Please don't forget this. This is very, very important. So, you have a cell secreting interferon gamma. This will act on macrophage. This will secrete. So, this is the cytokine that would stimulate interleukin 12. Just know the connection. Both will stimulate each other. 12 interferon gamma and interferon gamma 12. Okay. What's the answer? For isotype switching, what are the two requirements amongst what is given here? You need CD40 ligand and you also need interferon gamma. Interferon gamma also helps in isotype switching. So, antigen and interleukin 2 is not going to help in any way in isotype switching. CTLA4 and interleukin 4, CTLA4 has nothing to do with isotype switching. It's a suppressor molecule. B7, yes, but then interleukin 7 has nothing to do with isotype switching. CD20 and interleukin 13 has nothing to do with isotype switching. For isotype switching, you need CD40 ligand and you also need a cytokine. That cytokine could be interferon gamma, it could be interleukin 4, it could be TGF beta. It could be any of these cytokines, but CD40L is a must, plus a cytokine. Okay? Okay. So a T cell receptor has recognized an antigen, but it doesn't get any more signals. So what will happen? What will happen? Nothing will happen, isn't it? Nothing. Energy. It just stays like that. So this is what happens when a T cell is recognizing, T cell receptor is recognizing a harmless protein or a harmless peptide. No secondary signal, no co-stimulatory molecules, no interleukin 2. So just stay like that. Energy. This is what happens when our T cells are recognizing some harmless peptides. Okay? So no co-stimulatory molecules. The additional signal, signal 2 is co-stimulatory molecules, nothing happens. Signal 3 is interleukin 2. So unless this happens, this is not going to happen. So it just stays. Energy. Nothing. This one. Okay. Please read the question properly. What are naive T cells? What are naive T cells? They have never encountered an antigen. What does interleukin 2 do? Proliferation of activated T lymphocytes. So unless you have seen an antigen, you can pour interleukin 2, it's not going to respond. Okay? You can pour the interleukin 2 down the naive T cell's throat. It's not going to respond. Okay? So that's all what this question is asking you about, because you must remember interleukin 2 is the third signal after it meets with an antigen. Okay? So this, nothing is going to happen. The naive T cells, naive, they have not yet met any antigen, so they just stay there. Okay. It's an easy one. I'm not going to. Which one? Yes? Pardon me? Energy is after it's exposed to an antigen. Okay? After it's exposed to an antigen, and then there are no co-stimulatory molecules. The naive T cell is not exposed to an antigen. They have not yet they have not even, encountered an antigen. They are naive. That's a definition of a naive. Okay? Energy is the indifference after it has been exposed to an antigen. It does not want to respond. Okay? That's a difference. So this is, what's this? IgM. Okay. So the first T cell dependent antigen for the, whether it is T cell or T dependent or independent, it is IgM. But anyway, you know. Okay, so this is, let's see, let me just see whether you can guess. Which of these cytokines do you think would have an anti-tumor activity? The absence of what on a cell will help a cell to escape? What should be there on the surface of a cell? What should be there? MHC1, isn't it? MHC1 definitely on a tumor cell, they need that MHC1 to be identified and killed. Okay? Now, what happens is, the interferon gamma is a cytokine which can upregulate MHC1 and also MHC2 on many cells. So often when you want an upregulation of MHC1, they use interferon gamma. Okay, this I'm just letting you know because it's you should just know it. So interferon gamma is a cytokine that is often used to upregulate MHC1 and also MHC2 on the surface of cells. So the correct answer here is interferon gamma. So just yeah. Okay, but then interleukin 12, yes, it can. Interleukin 12 actually, okay, yeah, it can activate natural killer cells. But here the purpose is to upregulate MHC1 and MHC2 on the cells. So for that, interferon gamma is more efficient. Interferon gamma is more efficient for stimulating MHC1 and MHC2. So that's the answer. Okay, what's the answer to this? Isotype switch to IgA amongst the options given? Which one? TGF beta. Okay. Which one is this? A cell that secretes interleukin 4, 10 and TGF beta would be Th2. Okay. And this? Eosinophil. Okay. These are all rapid fire questions. Which one secretes interleukin 10? Th2 again. Th2. Which is the cell exposed to interleukin 12? Th1. Because it will secrete interferon gamma, which will activate phagocytic cell like macrophage. Okay? It's a good one. Read it. Okay, here, 1 second. I noticed this should be I'm sorry, I just noticed this. Okay, this should be absent. Okay, yeah. I was just reading and I realized that's a typo. So what is it? Huh? Yes, absence of CD40 ligand because there is no isotype switching. This child is producing IgM, but other serotypes, isotypes are missing. So what's this called? What's this disease called? It's called hyper IgM syndrome. Very common. Hyper IgM syndrome is when the CD40 ligand is missing. Okay, so this is another very commonly asked high yield scenario. Child has IgM. Yep. Absence of IgM. Normal numbers of lymphocytes bearing monomeric IgM are found by flow. Yes, yes. Monomeric IgM. Oh, this, what was, I did not see the monomeric IgM. I'm so sorry. I did not notice. I just thought, okay, if it is, okay, no, there was nothing wrong with the question. Okay, I'm sorry. I was reading it wrong. Okay, let's just go back. All the other isotypes are normal. Monomeric IgM is seen, which means that there is a deficiency of the J chain. Okay, I did not notice. I didn't even see the monomeric. I just thought IgM and I made a mistake. Okay, so wait, just hold on. Original question, monomeric IgM, all the other isotype switching is normal. That means there is a deficiency in the J chain, because it's a J chain that binds them and you get a pentavalent, yes, IgM. Thanks, I didn't notice it. And then if it was like how I've corrected, if you see only IgM, not monomeric, normal IgM, and the other isotypes are missing, then it's a CD40 ligand deficiency. Yes. Yeah, but you see, even if there is IL-4, the 40 ligand should be there. Yes, because what studies have shown is that even when there is cytokine IL-4, if CD40 ligand is missing, there is no isotype switching. Yeah. Okay. What would be this one? Phagocytic abilities is associated with which complement component? C3b. Okay, so this is C3b, opsonine C3b. Then so there is no secretory IgA, but IgA monomers, what is it due to? That was the last, I think almost the last PowerPoint I think I showed today in the class. Second last PowerPoint. What is needed for the dimer to cross? Poly Ig, isn't it? Yes. So you have the 2 dimer. Okay. So there is a poly Ig. This is required for it to cross the. Okay. So the correct answer is this. What's the answer to this one? You want to measure each class. So which antibody to which part will you use? What differentiates the different classes? Isotype. So you use an anti-isotype antibody. Okay? This is used for differentiating into classes. Okay, this, I don't know. So you have an immunoglobulin. Okay, this should be so what happens when you add pepsin? Okay, so you have an immunoglobulin like this. So when you add pepsin, what goes? This goes. Okay, when you add papain, you have like this, and like this. Okay, so which one can still bind to the antigen? The pepsin digested one can still bind to the antigen. Okay, so the agglutination can still take place. Pepsin digestion will still keep this intact. Okay, so the antigen can bind here. You can have cross react, you can have a lattice. This is completely gone with papain. Okay, pardon me? If it was papain, nothing will happen. There's no binding. See, with papain, all the functions of the antibody is lost. It just goes three parts, nothing. Okay, so the FC, this portion in a papain digestion, the FC, this portion can attach to a macrophage, but it doesn't serve any purpose. Yeah. Precipitation also, you need the antigen binding site. It has to be. Huh? Precipitation is usually soluble antigen. Yeah, soluble antigen and soluble antibody bind to form an insoluble precipitate. Yeah. Agglutination, RBC, antigen on RBC, it's agglutination. You see, it's a particulate cell. It is agglutination. RBCs cannot take part in precipitation. RBCs cannot take part in precipitation. Precipitation is a soluble antigen. When it combines with antibody, it becomes an insoluble precipitate. What's the answer? Remember today when I was telling you about the effector functions, I said IgM is an efficient. What is the IgM? Is it an efficient opsonizing antibody? Is it an efficient complement mediated opsonization? Or is it a cytotoxic or LAK? Complement activation actually. Okay, complement mediated. This is okay. This would be a more appropriate answer. So IgM is mostly complement activation, and IgG would be opsonization. Okay. Complement activation generally seen in, yes. Okay. This is a virus rubella. She had rubella during her pregnancy, and now the baby is born, and no, it's not born. Cord blood is connected, and you find that there is IgM and IgG. What is the most appropriate option here? Let's look at each one of them. Fetal and maternal infection occurred. Do you agree? Why? Maternal infection, it's anyway, they have mentioned it there. Why do you say that the fetal infection also occurred? Because yes, the cord blood had IgM. Okay? Now, infant produced IgG antibodies but not IgM antibodies before birth? No, that cannot happen. The infant was not exposed to rubella in utero? No, because IgM. Maternal IgM and IgG antibodies were both transferred? That also cannot happen because IgM won't cross the placenta. The infant produced both IgM and IgG antibodies while in utero? Yeah, no. The infant, okay, now in a long-standing infection, yes, it can produce IgM and IgG in utero. That is possible. But fetal as well as maternal infection occurred. That is definitely more appropriate because mother and both are infected, isn't it? Isn't that more appropriate than E? Yes. So that's why, yeah. Yeah, that's for polysaccharide. For polysaccharide vaccines, marginal zone B lymphocytes, yeah, this is nothing to do with that. This is a viral infection. Okay, this is protein. What's the answer to this? What kind of a response are they referring to? It's a secondary response. So predominantly IgG. IgG. So this is a secondary response. So you'll see IgG. So how do you make a hapten immunogenic? So what's the correct answer? Conjugation to a larger immunogenic molecule, which is a carrier protein. Okay, haptens, low molecular weight substances which can be tagged with a larger. So E is the correct answer. Okay, what about this one? What is an adjuvant? Enhances the immunogenicity of an immunogen. Do you think that 5 to 6 amino acid, a peptide, is that an immunogen? Do you think it's an immunogen? Just 5 to 6? No. Okay, so 5 to 6 is too small. Even though it's a peptide, it's too small to be an immunogen.